Okay, so for this unit, the recording has started, by the way, so if y'all want to mute yourselves now, please do. Um, so for this unit, what we're talking about is using DNA and genetics to help us solve real-world issues. So for instance, right, in the past 2 weeks, we've been doing labs over practical use of DNA. The first one was basically just telling us how we use it, right? Gel electrophoresis and PCR are ways that we can multiply DNA and actually use it. The second one, the elephant lab, was using genetic information to trace tusks to elephants and then those specific elephants back to their main groups. And that's a real world scenario in which we have to use DNA. And then in this week's one, which I hope all of you finished, we used what we were creating basically was a GMO, a genetically modified organism through a fly, right? So since we're talking about all that, y'all get the idea of what this unit is focused on, right? It's all about genetics and its practical use now. So let's get started. Pop open my whiteboard, clear all this off. So let's start with PCR. PCR stands for polymerase chain reaction. It's a chain reaction that we use to replicate certain segments of DNA in order to use them in a study, right? Because if I were to analyze all 46 chromosomes of a human, I would have way too much information. So when I'm trying to figure out who the father is, because I decided to go on the Murray show for a DNA test for some reason, I have to use very specific segments of DNA. By the way, I hope none of you ever end up on that show. I know most of y'all are 18 now, so just don't be stupid. Flip my computer around here. So PCR stands for the polymerase chain reaction, as I've already talked about, right? So when we're using this, right, we want specific sections of DNA. And in order for the reaction to work, let me draw an example. The DNA is supposed to be a double helix, I know, but for the sake of examples, this will be easier to understand if I just make it a ladder. So say, for instance, I'm trying to isolate this specific section of DNA. In order to isolate that specific section, first, I need to separate the double helix of DNA. In order to do that, I have to get it to denature. In order to do that, I have to do step one of the PCR process. I have to increase the temperature. The temperature will increase to about 100 degrees. Do it in red, right? Somewhere around 100 degrees Celsius, usually around like 97 to 99. And that is hot enough that the DNA will denature and all these bonds in the middle will split, which makes it go from being attached like that to having all of its hydrogens bonds split like this. And looking at that, we have two separate segments of DNA. Now, I'm still looking for these two middle sections here, right? They were once connected, but they have enough DNA information there that I can still use them. Well, the temperature is still around 100 degrees Celsius. And the next thing I need to do is begin transcripting that process, right? I need to replicate this DNA, right? So how am I going to do that? Well, first I need to create the environment, or I need to make the environment into something suitable for the process to happen. I got to decrease the temperature now. So the temperature will go from 100 degrees Celsius to about 58. All right, at around 58 degrees Celsius, what's going to happen is we have these little things called primers. I'm going to color them yellow. Actually, I probably shouldn't do that. Y'all can't see those. Orange, right? And these primers have a very specific set of bases to them. Those bases will attach to either here or here. Now, why is it on two separate ends? Well, remember, DNA runs in a 5' to 3' direction, right? I want whenever I attach that primer onto it, it's going to replicate DNA in the opposite direction, right? Granted, it's also going to produce a lot of excess DNA that I no longer need, but we'll get around to that when we get around to that. Either way, the main thing you want to know here is that I have to decrease the temperature in order to cause these primers to attach to the DNA. When those primers attach, it's going to look a little something like this. Just draw our lines. Oh, come on. Come on. My system hates me. It's being disagreeable. So now I have a primer attached here and a primer attached down here. Which means that the DNA is now going to replicate itself in that direction, right? Opposite directions, of course. And I'm still only looking for this middle segment here. So anything beyond that middle segment, like all of this, is just going to be excess DNA that I will deal with later, right? We'll actually deal with it. Don't worry, I'm not just brushing all this off. But so now that the primers are adhered, I need to get it to where I can attach a DNA polymerase and actually get this working, right? It's called the polymerase chain reaction. I gotta have polymerase on there somewhere, right? And polymerase doesn't work at 58 degrees Celsius. It works at around 73. So the last thing I need to do is bring up the temperature again. This time not around boiling, but around 58, or from 58 degrees Celsius to about 73, 72-ish, right? Some working temperature in which all of this stuff can replicate. And once I've hit that temperature, a big old enzyme called DNA polymerase is going to attach. We'll make it magenta because that's what we've been using as of late in our videos. It's going to attach, it's going to read the primer, and the primer is going to say, hey, start replicating, and it's going to replicate all of that. This is considered one cycle. The easy way to remember one cycle of the PCR process is how many times you change temperature. In one cycle, I change it once, twice, thrice. By the way, thrice is a word if y'all didn't know that. So every cycle, 3 steps happen, right? Now, in order for PCR to truly start working, I need to complete 3 cycles. So let's do another cycle, right? So let me erase most of this. So now what I have is after that polymerase does its first run through and replicates all that stuff that I need, it's going to have a lot of excess DNA. And the reason why you have to do 3 cycles is so that you can get rid of all that excess DNA that you're not actually using. If I can get all of this to, come on. Sorry. Oh, that's close enough. I can move the rest of it. Look at that. Technology working at its finest. All right. So after it all replicates, I'm going to have, well, a lot of stuff, right? So let me get rid of this. So after the RNA or DNA polymerase replicates its stuff, it leaves. And I have a bunch of finished strands. So let's color that in. I'll make I'll make this segment green so that y'all know this is the segment that we want. And everything else red. Right. All this red segment it's just extra DNA that we replicated, but we don't need. Now, the next thing I need to do is do the process over again, right? Because I have excess DNA that I don't need. So what happens next? Well, first the primers actually remove themselves. But now I need to do the process over again, which means heating up, cooling it down, and then heating it up again. So we do a plus 100 degrees Celsius, not exactly 100 degrees, by the way. It's just near boiling. And by doing that, it denatures, right? So I have my, what is it? I have my original strands. my necessary parts. So trying to draw this upside down is kind of hard. And then all my extra stuff. All right. So I want to get rid of the red part. I want to make it, or I want it to not be there, right? So once again, let me add in all the blue DNA. So once again, I cool the temperature down to around 58 degrees Celsius. And at 58 degrees Celsius, the primers can attach. So I have the primers attached. This time, the primers are going to attach here. Oh. Here. And oh, actually, let me move this towards the middle of it. You know what? I'll just redraw it. There we go. So next, I need another primer. So we'll get another primer on here. And then I have a primer down here like I did before. And a primer up here like I did before. And remember, here, I will color it green here as well. We're still looking for this middle segment of DNA, right? Now when it replicates, what it's going to look like at the end, after we increase the temperature back to 78 degrees Celsius, and we get the polymerase attached and it replicates the DNA, what it's going to look like is, well, this. We're going to have another copy of the excess DNA strand. So trying to visualize it. Hang on. This was played on an animation in class, if you don't remember it. Then at this point, I don't expect y'all to remember anything from like last week. So. All right. So first, I have a brand new replicated strand here. A brand new one here. One which has that. One which looks like that. And this one here has red DNA. This one here has red DNA. And these two in the middle had red DNA, right? So this one had it, this one had it. But after the primers replicate, what's going to happen is, since the primers were attached right here, right up against it, they can only replicate the green part, which means all I have left at the end of the second cycle is DNA with the green parts, aka the parts that I want. So this one, after I remove the primer, it's just going to have the desirable DNA that I wish for. Right here, let me erase the primer here. See that? Now, when I continue the chain reaction and I heat it up, I'm just going to keep replicating that green part, and it's going to increase exponentially, right? And the reason why we say 3 instead of 2 is because at 3, then we have enough of it that it can begin to exponentially increase. So in order to have enough workable DNA that I want, I would need to do 3 cycles of the PCR, just because at the end of 3 cycles, there's enough of this of these individual desirable DNA fragments that I can continue doing the cycle. All right, that's PCR in a nutshell. Any questions? Someone say no if y'all don't have any. Or type them in chat. I know y'all don't like talking. All right. Looks like we are good. So that's PCR out of the way. Now that I have a bunch of workable DNA, what am I going to do with it? Well, I already mentioned this earlier. Gel electrophoresis. What is gel electrophoresis? Well, it's what I'm going to put the DNA into, right? In order to do gel electrophoresis, I need all the DNA I made from PCR. So from PCR, I get, oh, my bad. I get a bunch of workable DNA, and I need to use it on something. So why is it called gel electrophoresis? Because it uses gel and electricity, right? And the purpose of this gel electrophoresis process is to separate specific alleles that we can analyze. Y'all remember alleles, right? It's the individual letters that we give to certain traits, right? Like if you're heterozygous and you mate with a homozygous person, right? You possess a dominant allele and a non-dominant allele, right? Or sorry, recessive allele, not non-dominant, my bad. So here's how we're gonna do it. First things first, I take all of my individual DNA and I put it into these boxes, right? And I'll make an example of a gel electrophoresis machine here. We'll call this side the positive, and we'll call this side the negative. So gel electrophoresis begins on the positive side because when the electrical current goes through it, electrical current will run positive to negative, and it'll cause the DNA, which I put into these beginning boxes up here, to run this way. It's like a racetrack, right? So let's put the DNA into boxes. First DNA segment I will use is my control, right? We'll call it con for short. And it'll have this initial box right here, and then everything else here measures specific DNA. And we'll say it looks like this. And da-da. Right. Now, we'll use red for my sample. And we'll call it SAM1 for sample 1. And it has a giant bank of DNA here. And then I'll run the current through it, and it looks like this. Ooh. And then I will do one more sample. We'll call it SAM2. Has a big bank of DNA up here, and oh, and like that. Okay, so let's look at this. What do the lines mean? The lines represent alleles, right? Each of these individual parts represents a different trait. What individual parts? These individual parts. This is one trait. This is one trait. This is one trait. Each like long separate segment is in and of itself a trait, right? Just look across, right? You'll see big gaps between the traits. That's how you'll know they're different. To properly analyze these things, you have to look at both the traits and how many alleles are in them. Well, how do I identify the alleles? Which surprisingly, the lab didn't tell you. Oh, someone. Did I run this in the opposite direction? I probably ran it in the opposite direction. Check your labs. But yeah, DNA is negatively charged, so if I remember correctly, hold on, I'm trying to remember. Actually, you know what? I can just check right now. Give me 1 second, guys. Ah, sorry, I did have it backwards. Yeah, change the direction. Oop. Sorry, this end's supposed to be negative. You put your DNA banks on the negative side. There you go. Thank you, Hamati, for correcting me. Smiley face. I appreciate it. Anyway, so yeah, sorry, my bad. Runs in the opposite direction. But still, each separate part is a trait. And how do I identify the alleles of each trait? I look at the lines. Each line represents an allele. And if there's only one line, like this one, that one, it's homozygous, meaning it has either two dominant alleles or two recessive alleles. Whereas this one, where it has two lines, it's heterozygous. It has one of each. That's it. I can't identify whether or not it's dominant or recessive, but I can identify whether or not it's homozygous or heterozygous, which is very, very important. Because if I know something is homozygous versus heterozygous, I can match it up pretty well. Not only that, I have multiple traits to analyze, right? So we could say that in sample one, this specific organism, for its first trait, is heterozygous, heterozygous. On its second trait, homozygous on its third trait, homozygous on its fourth trait, and homozygous on its fifth trait. That's how we would identify this. Now, why is it running all vertical, and why are there lines and all that jazz? Basically, each line represents the amount of base pairs, right? The bigger and higher your amount of base pairs there are in a specific trait, the slower it is, right? The easy way to remember is it goes big to small, right? And everything is measured in base pairs. So we'll say like this one is like 500 base pairs. And this one is around like, I don't know, we'll say, what's a weird scaling number? 250 base pairs. And these ones down here are only at 100. Basically, when you run the electrical current through the base pairs that of the samples that you collected through PCR, your bigger ones are going to run slower, meaning they stop closer to the beginning. Your smaller ones are going to run faster and longer, making them end here. Makes sense? Makes sense. So let's go back to actually analyzing this. Of my two samples, which one matches with my control sample? It's sample 2, because although it is a bit crooked, I meant to make this one match perfectly with the control sample. How do you know that two organisms are related? Well, you're going to have a test sample and you're going to have the organism that you got, and you just line them up. Like in experiment 2 or in lab 2, you were supposed to identify if a tusk perfectly matched with a specific elephant. All you had to do was line the lines up. Whichever one matched perfectly, that was it. And yeah, so when they're doing like genetics tests to determine whether or not you're the parent of the child, uh, since the child shares some of your DNA, they'll actually match some of your alleles on your base pairings. They'll combine that information with the information of the mother, and that's how they identify whether or not you're the dad. I hope none of you ever end up on Mari. That would be sad. But yeah, so that is basically all of gel electrophoresis. Then the last thing we need to cover is everything we did today. And all of that is vocabulary. So give me 1 second while I pull up my notes.